But now we're going to move on to the lightning talks. So this is a little fun session where we get one slide from each of the students who are participating. They get 3 minutes max to present on it. And if they start going over the 3 minutes, then a virtual kind of hook comes on. The slide will move forward and they'll be muted immediately. So anyway, we're going to do it in this order. We're going to start with Benoit, and then Julia and Dory, Mark, Sam and Lawson. And I'm going to say ready, steady, go.
Yeah, can you hear me fine?
Fine, Benoit.
All right. So thanks a lot for welcoming us to the Science Day, and I'm glad to be able to present my PhD project again, since it's my second Science Day. So I've refined my PhD project since the last time, and I will be focusing on generating molecule conformations with deep learning methods. Conformation generation is the field of trying to predict the 3D configurations of molecules. So starting from, for example, as you can see on the left, the topological formula. And there are various ways that are working quite fine to generate conformations using, for example, rule-based methods, which are relying on existing physics and chemical and chemistry rules, like molecular dynamics, quantum mechanics, or even distance geometry. And there are also a set of database rules relying on existing conformation data, for example, torsion angles profiles. Since it's quite impossible to perform a systematic sampling of all the existing torsion angles that could be existing for the conformations. It still, existing methods like the RDKit conformer generator, the confab or the omega, or even the CCDC conformer generators are working fine and retrieving most of the conformations that can be observed or could be determined using computational methods. But there are still some limitations, especially when sampling quite large molecules and, for instance, the macrocycles, which are cycles containing more than 9 heavy atoms. And there's been a quite popular trend in generating molecules using deep generative models, relying on SMILES or graph representations. And there could be an opportunity to perform the same in generating conformations with deep learning methods. There's especially an opportunity, for instance, to generate, to bias the conformational sampling into conformations of interest. And my project will be interested in the next weeks in trying to bias the existing deep learning conformation generators towards bioactive conformations. And this will be especially useful for pharmacophore modeling, or to try to have better input conformations in docking experiments.
Thumbs up.
Yeah, that's good.
Next.
Hello everyone. So say you're at a beach or on a snowy mountain and you're trying to make a sand or a snowball with your hands. You will use them to mold the sand and the snow in the same way, but the outcome is different. Whilst you manage to get a relatively spherical and compact snowball, your sandball has a disappointing croquette shape and it breaks down really easily. Well, the pharmaceutical industry has to go through a similar trial and error process when deciding which drug formulation is best for tableting. Every pharmaceutically relevant material compacts differently, and you can only find out how a blend of materials will behave by compacting them. So this is where my PhD project feeds in. The project is carried out at the University of Strathclyde in the SIPS department under the supervision of Ian Oswell, Daniel Markle, and Andy Maloney from the CCDC. The project aims to understand and predict how pharmaceutically relevant materials will function under the pressure conditions observed in the tableting process. This will be done by studying materials at a microscopic scale, so by tracking the slip lane movement in a system, and also at a microscopic scale. So this would be by measuring the elastic and plastic recovery of them, for example. Something I have been working on at the moment is testing hexamine as a pressure marker. So diamond anvil cells, as shown in the diagram, are used to study how pressure affects materials at a molecular level. To determine the pressure experience within the cell, you load a ruby chip into it and you track how far the R1 line of its fluorescent spectra shifts from its ambient pressure position. Ruby is a widely used pressure marker, but it has a low sensitivity at pressures under 1 gigapascal. This can provide inaccurate readings, so we tested hexamine as a more suitable gauge of pressure for this pressure range. We loaded the diamond anvil cells seen on the screen, so the circular one, with 4 ruby chips and a hexamine crystal, and we compressed it up to 1 gigapascal and decompressed it back down to ambient pressure 3 times as small incremental steps. We also took the Raman spectra of hexamine and the fluorescent spectra of the rubies at each step. The loading shown in the screen is not the one we used in the experiment, but it's so that you can have an idea of what it looked like. So looking at the graphs on the right-hand side, we saw that 2 distinct peaks in the Raman spectra of hexamine, one at 785 and the other at 1045 centimeters to the -1, shifted at increasing pressure in a linear manner. Additionally, the original Raman spectra at ambient pressure was recovered upon decompression. These are 2 very positive results. Therefore, the next steps now are to compare Raman shifts measured to the unit cell parameters of hexamine obtained through diffraction, and we will be able to link these to the pressure experienced in the cell through the equation of state determined by Charlie McMonagle in the paper cited below. Thank you for your attention.
Well done. 15 seconds left, so brilliant. And on to the next one.
Yes, so hello everybody. My name is Dori Gashparikova and I am a first-year PhD student in MK Pol's group at Durham University, and I'm also part of the MOSMED CDT. My project is titled From Fragment Screening to Fragment Growing, New Strategies for Drug Discovery, and it focuses on utilizing fragment-based strategies for the design of new ligands that bind and activate glucokinase. So the reason why we're focusing on glucokinase is because it is a very interesting enzyme that catalyzes the phosphorylation of glucose to glucose 6-phosphate, and therefore plays an essential role as a glucose sensor in the pancreas and in the liver. Additionally, in the liver, glucokinase is involved in glycogen synthesis because it aids the conversion of glucose into glycogen, reducing the glucose concentration levels in the blood. Mutations in the glucokinase gene are linked to type 2 diabetes, and type 2 diabetes is responsible for a large percentage of all diabetes cases. It is estimated to affect 150 to 400 million people worldwide. Therefore, identifying glucokinase activators has potential in developing drugs for the regulation of the glycemic imbalance that arises in people with type 2 diabetes. One of the conventional methods used for drug discovery is high throughput screening, or HTS. However, in recent years, using HTS has proven to be very costly and extensive, sometimes taking up to 14 years to create an FDA-approved drug. Therefore, alternatives, such as fragment-based drug design, are being utilized more and more. Fragment-based drug design is a method of developing potent drugs from molecular fragments or chemical compounds that have a low molecular weight and low binding affinity to a target protein. Fragment-based drug discovery includes fragment screening methods such as X-ray crystallography, docking, and surface plasmon resonance. Once fragments are identified, they're grown into drugs. In my project, I will be focusing on X-ray crystallography and using gold for docking to study protein-ligand interactions between glucokinase and a variety of compounds provided by Céline Cano's group in Newcastle to help predict the binding poses of these compounds. These compounds were identified in prior screening as potential activators, and I will be working to study their activity and molecular mechanism. This knowledge will then be applied to the design of other fragments and potential leads. When it comes to my lab work and results so far, I've been working on optimizing the protein expression and purification of a GST-tagged glucokinase. I've also been studying PDB entries of glucokinase and different ligands to identify which amino acid residues of the protein are involved in binding to the ligands and how they affect the binding pose. In the near future, I will be performing thermal shift assays to aid studying the effects of ligand binding to glucokinase. Following this, I will use fragment-based methods to find new scaffolds and other potential allosteric binding sites, which will lead to the development of fragment growing methods, probably sometime in my second year. That's it for me. Thank you very much for listening.
Perfect. Well done. 10 seconds left. So next.
Good afternoon, everybody. Computational approaches to structure-based design are prevalent in the pharmaceutical industry for the design of new drug compounds. The CCDC's cross-miner tool is extremely useful for identifying key interactions between drug and protein, as well as being able to search both the CSD and protein data bank in terms of customizable structural features to identify potential lead compounds. My work is looking at designing tools to expand the current functionality of cross-miner for the identification of optimal or new novel ligands for organometallic catalysts using a structure-based approach. To assess the activity of both currently used and potential ligands, I've developed a semi-automated, high-throughput computational methodology to calculate the activation energy for their derived active catalyst. Application of this workflow to 345 ligands used in the literature for the Ullmann-Goldberg reaction gives us a success rate of 91% for the generation of the correct transition states for 2 potential reaction mechanisms within 1 week. This is much faster than the currently used DFT methods. To enable easy searching of the CSD, I've also developed a structural database, CATSD, which is built upon the data contained within the CSD, but designed specifically for applications involving organometallic catalysis. CATSD contains key structural features used to describe ligand properties in the same manner that the default cross-miner database does for pharmaceuticals. Features include common coordinating functional groups, ring features, and common secondary interactions, which can be used to aid in the design and discovery of potential ligands from within the Cambridge Structural Database. Using CATSD with cross-miner will allow for searching the CSD using a catalyphore, which is what we've termed the pharmacophore equivalent for catalysis, which is seen down the bottom right here of the slide, which describes key features of the desired catalyst must possess to identify new or optimal ligands for a specific reaction. This, along with the accompanying computational workflow, will allow for the prediction of the resulting catalyst activity, enabling the development of catalyst synthesis in a faster and more efficient manner than was previously possible. So thank you for your attention, and if you have any questions, I'll be happy to answer those in the chat.
Brilliant. Well done, Mark. 26 seconds left. Indeed, if you want to ask questions to any of the students, please do just ask them in the chat and they'll respond to you there. Right, on to the next one.
Morning all. My name is Sam. I'm a PhD student at the University of Leeds, and my project is utilising data-driven methods to elucidate the structural origins of polymorphism. So polymorphism is a key issue across many different chemical industries. This is because different polymorphs of the same compound can display markedly different physical and chemical properties. This can make choosing the optimum form to take forward in things like a formulation exercise, make them quite expensive and a time-consuming process. Despite their importance and their costs associated, the actual origins of polymorphism are actually quite poorly understood at this point. And we believe that modern sort of data-driven methods, along with resources such as CSD, provide us with a unique opportunity to probe the CSD for structural features which can correlate to polymorphism. So key to the research that we'll be conducting is this concept of a polymorphophore. So polymorphophores, for those that are aware of medicinal chemistry, are analogous to pharmacophores, in that a polymorphophore has the ability to impart polymorphism to a structure when it is incorporated into the chemical backbone of another chemical structure. In this way, these scaffolds are sort of polymorphically privileged in that kind of way. We aim to use data mining techniques to generate a data set of the polymorphophores, and then use feature engineering techniques to identify the common structural features which are shared across this data set of polymorphophores to create a polymorphic feature set. This feature set will then be used to mine the rest of the CSD in order to ascertain correlations between the presence of the structural features and a compound displaying polymorphism. The most predictive of these structural features will then be combined to produce a mathematical model to predict polymorphism propensity. This will score compounds based on the presence of these structural features and we'll give you a score based on whether it is likely to display polymorphism and how many polymorphs, or the extent of to which it will display polymorphism. Finally, to validate this model, we aim to generate a library of novel compounds based on the Roy polymorphophore and screen them for their polymorph, and then use them to test the predictive power of this polymorphism propensity model. Thank you very much.
Hello, I think Jason lost his connection again. I think we can go to the last student, Layson.
Hi, so my project is focused on topological characterization of MOFs within the CSD MOF subset. And the way this is done is by taking 2 and 3 periodic nets, and you can also have interpenetrating nets, and you really want to simplify it into sort of the most simple form of connectivity to get this mathematical network. And what's really important is the way that the connectivity is preserved, because mathematical topology and sort of chemical structure topology is not quite the same thing, because the way atoms bond together, they can't sort of be manipulated to the same extent that you could do with a mathematical model. So what we're trying to do is find a simplified way of representing these structures, like MOF5, for example, which is in a primitive cubic net, to discover what the connectivity and the sort of like spatial layout offers. So for example, we could use it to find trends in mechanical properties or porosity or conductivity. So the way that we're going to do this is we can take the bonding information from CSD, which is not something that anyone does at the moment, and we can use that to simplify the structure and ensure that all the bonds that we have are correct. Because if you're using sort of like a third-party software and you're uploading a CIF file, all of the bonding information is not contained within that file, so it's all lost, and then you can get discrepancies where you'd have longer bonds or shorter bonds that should exist that don't exist, and then you might be left with clusters instead of a periodic structure, and you can get errors. And one thing we also want to achieve is some sort of validation technique, so that when you are using a software and you get given a topology for a structure, you know that it's correct. Because currently, if you're doing this using these software, you get a result, you just assume that it's right unless you're visually checking. And when you want to do a high throughput set, you know, it's going to take you a long time to look at 100 structures to check that the topology is correct, for example. And then obviously we can create some sort of web-based visualization tool so that you can understand and see more about structure topology for people who aren't so familiar with it.
Well done, Lawson. And that is the end. And that was brilliant. All the students there kept time. And thank you. And you can see they've got some really interesting projects going on across a wide variety of different scientific areas.